It's an honor to have the chance to talk with you about an interesting potential toxin and nutrient interaction and the impacts that this could have on a particular disease, neural tube defects in human babies. Although Dr. Keen and I had not coordinated ahead of time, we actually are discussing many of the same themes as far as maternal and child health, and also a history of knowledge about a particular toxin or nutrient. So fumonisin, the toxin that I'm going to be telling you about, is a mycotoxin, and I recognize that that may not be a familiar term to many of you. So first I'll give some background on mycotoxins and their impacts on human health. And fumonisin is one example of a mycotoxin that has caused all kinds of problems to public health. As long as we've known, I'll describe what it is and what the postulated human health effects are. Then I'll describe folate and its role in the prevention of neural tube defects as far as the epidemiological evidence, give a little bit of discussion on folate-fumonisin interactions potentially in reducing the risk of neural tube defects, and then finally, what can we learn from this potential interaction as far as how we might change our paradigms for risk assessment? And although there's not a great deal of evidence, so I'm already kind of giving away the main point as far as the folate and fumonisin interaction, I'd like to present some ideas as far as a viral and toxin interaction. And the specific one that I'll be describing is the interaction of aflatoxin and chronic infection with a hepatitis B virus in causing human liver cancer. So first, what are mycotoxins? These are chemicals that are produced by the fungi that colonize our food crops. If any of you have ever done any gardening and you've seen that there's insect damage on your tomatoes or your corn or some other crop, and then you see this ring of mold around where the insect damage has occurred or where the shell or kernel has cracked, well, there certainly is fungal infection and possibly mycotoxins in that foodstuff. These mycotoxins can cause a variety of adverse toxic and carcinogenic effects depending upon the fungus that has produced the toxin as well as the substrate, that is the particular food crop. There's a fairly long history of mycotoxins affecting society. As far as the literature goes, this goes all the way back to the third book of the Bible, Leviticus. If you look in chapter 14, verse 37, you'll see that God issues an edict to the Israelites to cast the moldy stones outside your city walls instead of using them to build houses and other public buildings. So even then, thousands of years ago, there seemed to be some public health knowledge that mold exposure was not good for human health. Now fast forward to the 11th century. In medieval Europe, the main grain that was used to produce bread and cereals and porridge was not yet wheat, and it certainly wasn't yet corn or maize, it was rye. What you can see in the upper right-hand panel there is rye that has been infected by the fungus Claviceps purpurea. Claviceps purpurea produces very potent toxins, erga-alkaloids, in rye, and you can tell by that the grains are a little bit dark and shriveled in that picture. Well, this moldy rye was the cause of a disease known as St. Anthony's fire that was a problem throughout medieval Europe, and St. Anthony's fire was actually diagnosed as late as the late 1800s in Europe from consumption of moldy rye and rye products. This disease was called St. Anthony's fire because it was believed that taking a pilgrimage to the shrine of St. Anthony in present-day France would cure humans of this disease, which was characterized by essentially shriveling and blackening of the fingers and toes, hallucinations, gangrene, and eventually death. When people made a pilgrimage to the shrine of St. Anthony, they were indeed cured of their symptoms. And the reason for this was that actually in that particular parish, farmers were ordered to sieve out those darkened grains of rye, not to use them to produce flour for bread and for porridge. And this was primarily for religious reasons, but it actually turned out to be a pretty significant public health benefit as well. So people would take the pilgrimage, they would be cured of their symptoms, say hallelujah, go back home, they would get sick again. Now, fast forward again another 1,000 years roughly, during the Great Depression era in the United States in the 1930s. And there was also a massive drought throughout the Midwest during those years. And we actually used to be planting a lot more wheat in the United States than corn, and now we're planting a lot more corn than wheat. And part of the reason was because of that massive drought, because corn is relatively more drought tolerant than wheat. Unfortunately, under drought conditions, corn becomes very susceptible to contamination with a fungus, Fusarium verticillioides, which produces the mycotoxin fumonisin, which you'll be hearing more about today. Fumonisin has been definitively linked to a brain disease in horses, equine leukoencephalomalacia, and many horses in the 1930s were dying of this mysterious disease that was known as staggers. They'd be staggering around and they would drop dead within a few days of the onset of symptoms, and nobody understood why at the time. Also during the 1930s, anecdotally, there were many babies being born with neural tube defects in the United States, and again, nobody understood why. Now, today, we believe it is because of increased fumonisin exposure during that Great Depression era. In fact, it wasn't until 1960 that the first mycotoxin, aflatoxin, was discovered in connection with over 100,000 turkey poults dying in the United Kingdom from consumption of contaminated peanut meal. It had very, very high levels of aflatoxin. Now, today, there have been several dozens of mycotoxins identified and characterized, and one of them is fumonisin. Fumonisin is actually a class of mycotoxins that all have the same long-chain hydrocarbon backbone with some different R groups attached. Fumonisin is a mycotoxin produced primarily by the fungi Fusarium verticillioides and F. proliferatum. They infect a variety of food crops, although most commonly fumonisin is found in corn. It's also found in sorghum, to a limited extent in rice, a few other crops, but almost entirely in corn. And this is primarily in corn that's grown in warm or in temperate regions. So until the late 1990s, fumonisin was a problem pretty much every year in corn in the United States. It was actually the advent of transgenic Bt corn and the fact that we're planting a lot more of it today because it's protected against insects that our fumonisin exposure has gone down dramatically in the United States. The possible human health effects are esophageal cancer, childhood stunting, and neural tube defects, and I'll be describing some of the epidemiological evidence and the lack of evidence for each of these health effects. Around the world, the most highly exposed individuals are those that consume the most corn in these warm and temperate regions, particularly certain populations in Latin America and East Africa. So what is the mode of toxicity of fumonisin, and why are there such diverse effects? Well, fumonisin has the impact of affecting nutrient transport across cell membranes. Fumonisin very closely resembles the molecular structure of sphingonine and sphingosine, and these are the two molecules upon which the enzyme ceramide synthase acts to produce what are called complex glycosphingolipids that help to transport nutrients, including folate, across cell membranes. And when essentially when ceramide synthase is inhibited by fumonisin, then these sphingolipids are depleted and there's decreased nutrient uptake by cells. Now, this can cause a variety of adverse health effects, and one of the postulated ones in human populations from being exposed to high levels of fumonisin in corn is esophageal cancer. There's about close to 500,000 cases per year worldwide, of which there are two main types of esophageal cancer. The one that's the most common worldwide is squamous cell carcinoma, which is a cancer of the cells that line the esophagus. And then more common in the United States is adenocarcinoma, that's more associated with reflux and type 2 diabetes and obesity. This is a cancer of the cells that produce mucus in the esophagus. Now, the risk factors for one of these types of cancer, squamous cell carcinoma, are alcohol consumption, tobacco smoking, drinking hot drinks above roughly 170 degrees Fahrenheit, and possibly fumonisin exposure. Whereas, as I mentioned, the risk factors for adenocarcinoma, again, are smoking and gastroesophageal reflux disease and obesity. So what is the evidence that fumonisin might be associated with esophageal cancer? Unfortunately, all we have, to go back to Dr. Coates' earlier slide as far as the goodness of epidemiological studies, all we have in this case are ecological studies that have been conducted in China, Iran, and South Africa. So they're somewhat suggestive of an association between fumonisin exposure and esophageal cancer, but not really very conclusive. Two studies conducted in different counties in China seem to show that in the one county, Huai'an in Jiangsu province, which is just north of Shanghai, fumonisin exposure is extremely high through moldy corn, and also esophageal cancer incidence is extremely high there, over 80 per 10,000 in the population. Whereas in other populations where there's lower imputed fumonisin exposure, then the esophageal cancer rates are significantly lower. However, it's worth noting that in one particular county, Fusui, in Guangxi province in the south of China, where there's very high aflatoxin exposure, there's also very high liver cancer incidence. Now, as far as the studies conducted in Iran and South Africa, again, these are ecological studies. In the Iranian study, maize was simply sampled from two different areas, Mazandaran and Isfahan, and it was found that the maize in Mazandaran had extremely high levels of fumonisin, and indeed, esophageal cancer incidence was higher in Mazandaran than in Isfahan. But the esophageal cancer incidences were not given, and moreover, other confounders and cofactors were not taken into account in this particular study. And similar results and similar potential weaknesses in a study that was done in South Africa that showed that fumonisin intake was much higher in Kentane than in another area, Bizana, in the former Transkei region of South Africa. And indeed, esophageal cancer incidence was higher in Kentane than in Bizana. But again, the esophageal cancer incidences were not given, and the confounders and cofactors were not taken into account. And I think it's particularly important to take that into account in this study in South Africa, because the main source of exposure to fumonisin in the former Transkei region of South Africa is alcohol, a kind of beer that's made from moldy maize. So is it the fumonisin or is it the alcohol consumption that's leading to esophageal cancer? We don't really know, and that has not been teased out. Now, what about the link between fumonisin exposure and childhood stunting? Stunting is defined as a condition in which a child's height for his or her age is 2 or more standard errors below what the World Health Organization considers an acceptable height or a growth reference. That is, the child's height for age z-score is less than or equal to -2. It's not so much that shortness in and of itself is harmful to human health. Rather, that stunting, particularly if mediated by environmental conditions, has been associated with cognitive impairment and an increased vulnerability to infectious diseases. What you can see from this world map on the right is that the areas that are highlighted in dark red in sub-Saharan Africa and South and Southeast Asia are areas where the stunting prevalence is the highest among children under the age of 5. Worldwide, there's close to 200 million stunted children, and there are multiple different causes. Genetics is one. If you had short parents, you're more likely to be short as well. Malnutrition, infectious agents, and maternal factors such as how long the mother has breastfed her child, or does she happen to give particular foods more preferentially to boys versus girls, or older children versus younger children, etc. Now, what is the evidence linking dietary toxins to stunting? A lot of work has been done relating aflatoxin to stunting, and now there's been this recently published study and another one coming out soon relating fumonisin exposure to childhood stunting as well. In this particular study that was carried out in Tanzania, this was a study of 215 infants, and fumonisin exposure was estimated by measuring the amount of fumonisin that was present in the maize flour in these villages in Tanzania, and then asking mothers, well, roughly how much of this flour did you give to your infants over the course of a year? At the age of 12 months, the 26 infants whose fumonisin exposure calculated through this method, those who exceeded the JECFA tolerable daily intake of 2 micrograms per kilogram body weight per day were significantly shorter and higher than those infants who were consuming fumonisin at levels below the tolerable daily intake. And what I liked about this study was that they actually did adjust for confounders and cofactors, total energy intake, sex, village, and protein intake. And as I had mentioned, there's another study coming out showing similar results, so the body of evidence is growing. Now for neural tube defects. These are embryonic defects of the brain and the spinal cord in fetuses in which the neural tube fails to close fully in utero. The two most common neural tube defects worldwide are spina bifida, in which the spinal column doesn't close, and this can lead to nerve damage and paralysis that lasts throughout a lifetime, and a more severe condition, anencephaly, in which the brain doesn't develop, and you can see this in the picture of the baby above, that in this case the baby has anencephaly, and the result is either a stillbirth or death shortly after birth. Maternal folate consumption is absolutely crucial to reducing the risk of neural tube defects. And now this is an interesting case because fumonisins actually, they disrupt sphingolipid metabolism as I described earlier, and this hinders folate transport across cell membranes. In fact, it's been shown in a study by Janae Jellinal-Van Wees and her colleagues at University of Nebraska that fumonisin has been shown to induce neural tube defects in mice. And a variety of studies followed this one that replicated those results. So what is the link between fumonisin exposure and neural tube defects in babies? A variety of ecological studies have been done, but I want to focus on this one particular case control study because this is the highest quality of the epidemiological studies, conducted by Mismer et al. Along the Texas-Mexico border, published in Environmental Health Perspectives in 2006. In this particular case control studies, mothers were chosen because they either had or did not have babies with neural tube defects along the Texas-Mexico border. In this particular group of mothers, there was very high maize consumption. This was a primarily Hispanic American population. Very high maize consumption, and a lot of the maize that was consumed was in the form of corn tortillas that had not undergone any sort of regulatory inspection for fumonisins. Here in the United States, we have the Food and Drug Administration has set industry guidelines for allowable levels of fumonisin in a variety of different corn products for both humans and for animals. So how exactly did they measure fumonisin exposure in this case? Through three methods. They asked the mothers to estimate roughly how many tortillas they consumed every day during the first trimester of their pregnancy. They took blood samples and measured for serum sphingonine and to sphingosine ratio, which is it's an unvalidated biomarker of fumonisin exposure in humans, but it's been shown to be a pretty good biomarker of fumonisin exposure in various animal species. And then they also imputed fumonisin intake by looking at how much fumonisin was in the maize and estimating how much the mothers consumed. And the results of this study showed that there were, so if you could plot odds ratio actually along the y-axis and fumonisin exposure along the x-axis, that there were increasing odds ratios of risk of the mothers giving birth to babies with neural tube defects with increased exposures of fumonisin, except at the very highest dose of fumonisin. In that case, there was the lowest odds ratio of giving birth to a baby with neural tube defects. And the authors explained this by saying, well, maybe fumonisin exposure was so high that the babies were actually just dying in utero. The mothers actually never gave birth to them, although we certainly can't prove that. What's also an important point is that in this particular population, there was also very low dietary folate, and that could have also been a really important risk factor. So now turning from fumonisin to folate. There's the molecular structure of folate. This is a naturally occurring vitamin that's found in certain foods, and I'll give a slide of that later. The term folate is for the compound that's found in food, whereas folic acid is the synthetic version of folate, and that's been added to certain fortified cereals, for example, or bread, and also is found in multivitamin supplements. Now, humans need folate in order to synthesize, repair, and methylate DNA, and it's especially important that we get adequate folate when in periods of rapid cell division and growth. Hence, folate is really important as far in the fetal and the infant stages of life. Folate has also been linked to protection against anemia, heart disease, and certain cancers, particularly colorectal and prostate cancer. But the weight of evidence is especially strong for folate's role in the prevention of neural tube defects. Okay. So this is a list of some folate and folic acid-rich foods, and if you take a look down this list, as I'll be describing later, even in high-income, middle-income, and low-income countries, we don't necessarily get a lot of, we don't consume large amounts of some of these foods that are high in folate. But now, of course, if we consume breakfast cereals and breads, then we are getting folic acid in our diet. Now, what the top map shows are the countries of the world that have mandatory programs to supplement folic acid in breakfast cereals and in breads and other grain products. So those are the ones in the dark blue. The ones in the medium colored blue have voluntary programs for folic acid supplementation. Those in pale blue have planning programs, and those in white have no program activity for supplementation with folic acid. Now, what you see in the lower graphic is the evidence for neural tube defect rate decreases in the United States. Fortification with folic acid was introduced in the US in 1996, and then it was mandated in 1998. And what you see in the Hispanic, non-Hispanic white, and non-Hispanic black populations is that generally you can see a decrease in neural tube defect rates per 10,000 live births in the United States since the time of introduction of fortification with folic acid. Now, if you were to take a look around the world, this graphic shows 5 countries that have introduced folic acid supplementation. The U.S. in grain products, the U.S., Canada, Chile, Costa Rica, and South Africa. What you see in the blue bars are the rate of neural tube defects per 10,000 live births before the folic acid supplementation, and the green bars show the rates of neural tube defects after fortification with folic acid. So you can see there's some pretty strong evidence linking folic acid supplementation to lower rates of neural tube defects. Now, what is the role of folate in prevention of fumonisin-related neural tube defects? I would say that the evidence is unclear at best. Because fumonisin is a relatively recently discovered toxin, it was discovered in 1988, and we do not actually have a validated biomarker for fumonisin exposure at this time. So in these populations where the folic acid studies were being done as far as the reduction of NTDs, fumonisin exposure was not being measured at the same time, so you don't really have these 2 risk factors being looked at together in epidemiological studies. Moreover, we need to remember the mechanism of toxicity of fumonisin, that it inhibits folate transport across cell membranes. So if you keep on adding more and more folic acid or folate to the diet, does it do any good if the fumonisin is preventing it from crossing cell membranes? And as far as the existing animal studies, they have conflicting results. So the Jeleno van Wees et al. Study in 2005 showed that if you administer fumonisin to mice and then you supplement with folic acid, then you reduce the risk of neural tube defects. But those same authors actually just published a study showing, attempting to do the same type of study, and they actually found no protective effect of folic acid supplementation in mice that were fed very high levels of fumonisin. Well, that's not very good. So we don't really have the quantitative evidence there yet. Nonetheless, there is more evidence for the interaction of certain toxins and nutrients that may mean that we need to rethink our risk assessment paradigm. And here I will make a case for that. We might have some insights from viral and toxin interactions as to how that risk assessment paradigm may change in terms of looking at nutrients and toxins together. The example I'll give very briefly, because I know my time is running out, is that of the synergism between aflatoxin exposure and hepatitis B virus in causing liver cancer. And specifically in that 4-step risk assessment that you've heard all the speakers talking about earlier, what would be changed in this case is the dose response assessment. How would that be changed? It could, so specifically for the case of aflatoxin and hepatitis B virus working in synergy, we would change the slope factor or the cancer potency factor because we're evaluating a carcinogenic effect in humans, that is liver cancer. But for other nutrient and toxin interactions or virus and toxin interactions that have non-carcinogenic effects, what we might be changing is the NOEL, the no observed effect level, or the benchmark dose, the point of departure from which we can calculate reference doses or tolerable daily intakes. In the case of aflatoxin, this is another mycotoxin, by the way, produced by Aspergillus flavus and Aspergillus parasiticus, again in corn, and in a variety of different nuts, such as peanuts and tree nuts like almonds and pistachios and hazelnuts. Again, exposure is highest in the warm regions of the world where corn and peanuts are dietary staples. So again, sub-Saharan Africa and South and Southeast Asia, but it's also perennially a problem in the South and Southeast United States in the crops we produce there. The main health effect of interest is liver cancer. Now, when we tried to do a risk assessment to say what the global impact of aflatoxin was, we needed to use two different cancer potency factors. For those individuals who are exposed to aflatoxin but are not chronically infected with hepatitis B, and for those who are infected with hepatitis B, which is close to 500 million people around the world, because for them, actually, aflatoxin is a lot more dangerous. It raises the risk of liver cancer by up to 30 times. So, and this was something that was established by the Joint Expert Committee on Food Additives back in 1998. So we had to conduct the risk assessment separately for the populations of the world in different nations that were HBV positive versus HBV negative. We managed to capture a population of close to 6 billion people, and we found that there were roughly up to 155,000 cases of liver cancer per year caused by aflatoxin, but most of those cases were actually in individuals who were already hepatitis B virus positive. In fact, if you can get rid of HBV, then actually the impact of aflatoxin on global cancer risk decreases dramatically. Can a similar approach be used for nutrient toxin risk assessment? That is, you have completely different cancer potency factors depending on whether individuals are sufficient or insufficient for intake of a particular nutrient. Well, not necessarily for folate and fumonisin because we do not have the quantitative data, but what about for other examples, such as populations that are sufficient or insufficient in folate and cobalamin and the toxic and carcinogenic effects of arsenic? There is more evidence along those lines. What about for individuals who are sufficient in isothiocyanates that are found in cruciferous vegetables, having a much lower risk of aflatoxin-related liver cancer? That's something else for which we have more evidence. In these cases, we could calculate two different slope factors for the carcinogen based on the adequacy or lack thereof for the nutrient in that population that counteracts those effects. And you might have other examples of these nutrient-toxin interactions for which you might have completely different NOELs or BMDLs or cancer potency factors. So recommendations for future studies. We need more epidemiological studies on how fumonisin exposure contributes to neural tube defects in babies worldwide, and these toxicological and epidemiological studies that study both folate and fumonisin together, and whether the folate can actually reduce neural tube defect risk in those populations that consume high amounts of corn with fumonisin. And more broadly, determine if enough evidence exists to justify different dose-response assessments for nutrient-toxin interactions in populations. Thank you very much.